Hello, everyone, and thanks for joining us. I'm your host, Jenny Morganwick, and welcome to another edition of Jax Live. Today we have Kat Lutz joining us, who is our Senior Director in In Vivo Pharmacology and also the Mouse Repository. And we're going to talk with her about her group's work in preclinical studies using mouse models of ALS. But before we get going on the interview, I do want to follow up with some housekeeping. I want to let the audience know that you can use the comment box on whichever platform you're joining us from. You can ask questions, leave a comment, give us a shout out, let us know where you're tuning in from, and we'll take your questions in real time as we can. And if we get a bunch of questions, we can address them at the end of our chat with a live Q&A. So make sure you stick around for that. So Kat, thank you for joining us again. We've had you on before to talk about the HAS2 mice, and now I want to pick your brain about ALS. So welcome back.
Well, thank you. It's nice to be here today.
Yeah. Full disclosure, everyone, I work very closely with Kat, but a lot of our conversations span many different areas. So I'm excited to learn more about, what her group has been doing with ALS and particularly in the preclinical space. So before we get into that, though, Kat, I do want to know more about your work with neurodegenerative diseases in general. So I know you have extensive knowledge and experience with these, as well as rare diseases. So can you talk a bit more about areas you've worked in and how they might complement the work that your group is doing with ALS?
Sure. Well, I've always been a mouse geneticist at heart. In graduate school, I cut my teeth on epilepsy, and so I worked in areas of absence epilepsy and mouse models of absence epilepsy. If we fast forward many, many years into the era of spinal muscular atrophy, I will say I think that was really the disease for me that sort of framed what I feel is a successful roadmap or an infrastructure, if you will. I think it really starts with the mouse models, making sure that we have good mouse models. Most importantly, making sure they're available to the scientific community. Spinal muscular atrophy is a neurodegenerative disease. It's a motor neuron disease. It very much mimics some of the disease progression of ALS, but more horrifically, it happens in young children and in babies. And so the counterbalance to SMA and ALS is that SMA is the most common form is a single gene mutation. And so our ability to model that disease in mice was I'm going to say, maybe a little bit more straightforward because it was only one gene that we were looking for. But we did have challenges in modeling the various forms of SMA. I'm happy to say that, SMA is one of the biggest success stories, I think, in neurodegenerative diseases, with many FDA-approved therapies out there now. I believe there's 3 with Roche coming online. And so I think it is a really good success story. And again, at the heart of that success is the infrastructure and the resources that help you get to those therapeutics. And we're really hoping that with ALS, we can use that same sort of that infrastructure to try to make some progress into therapeutics for this disease as well.
So speaking of ALS in particular, what are some major advantages and challenges that you have experienced with the mouse models so far?
Yeah, I think I think ALS is rather challenging. Unlike SMA, it's a very complex disease. Most of the forms of ALS are what we call sporadic, which essentially just means that there's no evidence of an inherited form, within a family. So with the majority of ALS being sporadic, you're at a acute disadvantage. We do have genetic forms of ALS, and I think most recently we've had, some great successes in the gene identification, which has helped us model. But before we had genome sequencing, I'm going to say there was just a huge dry spell by which we only knew a couple of genes that were causative and involved in ALS. And so most of our research, in the past 20 years have been done on a very limited form of the disease. And that's been rather challenging. We will say also that, the forms of the disease that we have been able to model in mice, require somewhat of an artificial overexpression at times, SOD1 being the most notable. And so, models are only models. They're only as good as you can make them. And so we have to take with a grain of salt the information we get from them, and then how we translate that information into potential therapeutics.
So you mentioned, genome sequencing, and I know that the genome sequencing of both the sporadic and familial ALS patients was a huge breakthrough, as you alluded to, for ALS, since it allowed the identification of genes that could contribute to the disease. So could you kind of maybe go into a bit more detail as to how that has particularly helped translate into preclinical ALS investigations, or particular new mouse model developed, kind of the information we got from that genome sequencing?
Yeah, I'd be happy to. I think the genome sequencing of patients really opened up the door for the identification of genes involved in ALS in an exponential fashion. We had such limited knowledge before, and again, maybe focusing all of our efforts and our thoughts on cytotoxic, excitatory toxic pathways, which are not a bad thing to focus your attention on. But there are other pathways that have been uncovered. We know a lot more about ALS, the genes involved in ALS, and RNA metabolism and axonal transport. There's these groupings now that are starting to appear as a result of knowing what the underlying function is of all of these genetic diseases. And I think it just, expands our viewpoint, everything from axonal transport to RNA metabolism, to microglial activation. These all become new areas of investigation that have been really exciting over the years. And hand in hand with the genetic identification has come genetic engineering efficiencies, and I will say breakthroughs, if I could, to really help us understand how to rapidly make those models and to phenotype them and to understand exactly what they can contribute to our overlying and underlying knowledge, I should say, of this disease. So all of those genetic forms, even though there may be 10% of all the forms of ALS, are going to provide us with a much better vantage point of the mechanisms of the disease that I think we're going to see in the sporadic patients as well.
Yeah, that definitely does make sense, thinking about the disease. We do have a great question from the audience. So we have a guest that would like to know if you could tell us about a specific genetic model that your group has a lot of experience with, or and what are some of the caveats of working with that particular model? So maybe this is a good time to hear a bit more about one of your favorite specific models that you work with.
Well, it's interesting, when we say that there's a model of ALS, a lot of people like to see that it checks all the check boxes, right? That it has the motor neuron degeneration, that it has, the paralysis, that it has a lot of the maybe various forms of something that mimics a bulbar onset versus a limb onset. And so I think we've tried really hard over the years to look at that. But, C9orf72 was one of the mouse models that we started working with really early, as soon as that gene was discovered, because it does represent, so much of the familial form of ALS. And it's a repeat expansion disease. And interestingly, when you have a transgenic model that has a high repeat expansion, you don't necessarily get right away all of the physiological paralysis and various forms of that particular parts of the disease, but you demonstrate really nicely this recapitulation of the pathology of the dipeptide repeat expansions. So these dipeptide repeats accumulate, and the pathology, you might think of it a little bit like Alzheimer's and beta amyloid. It's one of the hallmarks, for that disease. And so even if we could get therapeutics that could potentially target those DPRs, it's an example of when maybe the mouse model isn't perfect, but maybe it's good enough, to investigate these particular areas that you want to look into. I think the other thing about the C9orf72 model, both the transgenic and the knockouts, is there is a clear emerging theme of environmental components that are really influencing this. And we see this, of course, in the human population. Part of the reason this disease is so hard to get your arms around is because we don't know what causes it, but epidemiologically, we know that there are environmental risk factors, if you will, being in the military being one of them. And so it becomes really interesting now to investigate the microbiota of the mouse, the environmental influences, to start thinking about exposures to viruses and common aspects from the human component, and then thinking about not just genetically engineering mouse models, but also trying to recapitulate that environmental exposure as well. So I think we have a long way to go, but some of these newer models I think have been really quite insightful.
So sticking with the theme of the C9orf mice that you just spoke of, so in lieu of SFN this year, JAX is going to be hosting 2 scientific sessions. They're going to be the same session on October 21st and 28th, so it gives our audience a couple different options of attending. And as you know, members of your team will be presenting at these sessions on new biomarkers in preclinical studies for ALS. So can you give our audience today a high-level overview of what members of your team will be talking about in these sessions?
Sure, I'd be happy to. One of the things that we do here at JAX, and it was one of the things that we did, I think that was quite impactful for SMA, was that we didn't have people reinventing the wheel. We had the mouse models for SMA here, and we were very, very familiar with them. And so JAX offers, this great infrastructure of really large vivariums, really large colonies of mice. And so the hurdles, the energy of activation to use these mice is lowered when we're working together in these disease areas. We're able to get mouse models of ALS and SMA and many other diseases as well up and going. We have all of the in-life assays, whether they're behavioral assays or whether they're histological assays. We know them, we know how to work with them. And so in working with the scientific community in order to help with their drug therapy studies so that we can get these answers, as quickly as possible. So when we work with these mouse models, we're trying to find aspects of the model that are clinically translational to the disease. And so one of them is electrophysiology. We have a lot of mouse models that exhibit, some degree of paralysis, which could be relatively subjective. So the SOD1 G93A will develop progressive paralysis. But what Laurent will be telling you about is his work with CMAP, the compound muscle action potential, which is exactly what they use in the clinic to measure the progression of ALS. And so not only can we look at the progression of ALS in these mouse models, we can do it much earlier in the disease because this technique is so very sensitive. And then, likewise, just because you have a mouse model, you really have to have the reagents, around them to be able to study them. And I think that we've been particularly challenged in some of the reagents that we have in terms of antibodies and histological readouts. And there's been a lot of people who have been, really trying hard to, just really kind of tackle these problems. And so again, I think what we offer, here at JAX is not only the mouse models, but the resources to be able to help you work on those areas so that you don't have to reinvent the wheel in your own lab. We can help with those kinds of studies.
And that's great because we just got a question from someone in the audience that says, I think we tend to rely on functional behavioral endpoints too much, although the clinical trial endpoints are of this sort. What do you think about more proximal endpoints to the targets in ALS? And you touched on that with CMAP. Do you have any other comments on that, more proximal endpoints?
Well, I think the biomarker, data is going to be important. Developing that biomarker data, not just in the patient population, but looking at it translationally as well. Not all ALS is the same, and we know that. And so to the best extent that we can stratify the patient population into various maybe RNA-seq upregulation, downregulation of certain pathways, the better chances we'll have of enrolling them into therapy that's targeted along that line. Same sort of comment on the clinical trials is that not only do you need the biomarkers, you really do have to demonstrate target engagement of your compound. And I think that those two things, are something that have been a major focus of improving clinical trials. And I think that's, really where our emphasis should be and is. So I think that that's good news. And then trying to find those same sort of biomarker data and analyses in the mouse models and see how they can match up. And then I think finally I'll say that I think we can learn a lot from iPS cells, both from patients and also engineered on coisogenic backgrounds. We really can look for specific mutations that we can CRISPR, for example, into an iPS cell line and look to see what exact pathways, those would be altering. So I think, the omics data, if you will, is really going to help inform our clinical trials as well.
So the last question I have for you, I would like to know, what is one of the most exciting things, in your opinion, to come out of preclinical work being done with ALS? I think you've covered a lot of exciting things that are generally interesting to everyone that's studying this, but I want to know what's getting you excited.
I think a lot of times when we think about tackling big diseases and big, I mean complex, big complex diseases like Alzheimer's and Parkinson's and ALS, I think you have to, you have to focus on your short wins as well. We don't have an overall therapy or a cure, today, but we have had, good short wins along the way. We have had Riluzole, we have had Edavarone, and now we have a new compound that has shown really promising phase 2, phase 3 results in the clinical trials by a company called Amylyx. And I like that because it really was a compound that worked in the ER and mitochondrial support so that it slowed the progression of the disease down. And so, just like a lot of the diseases that we're working with today, you may be looking for the cure, but along the way, you'll find the impactful therapies. And I like the genetic models of ALS because I do think that what we know about them, and sometimes it kind of goes to, is this good enough? Where if you have SOD1 G93A dominant gain of functions, if you have C9orf72 repeat expansions, the therapeutics designed to target those very specific genetic defects, and some of the clinical trial results that we've seen so far have been absolutely astounding. And it's just something as simple as, just removing, that toxic, gene. And now you say it's something as simple as, now, of course, there's some complexities to it, but it's one of the most straightforward pathways, that we have in these genetic models. And I do believe that the genetic models that we have really will inform the sporadic cases and the more complex, cases that we see. So I'm really excited about, seeing how some of those therapeutic strategies, those disease-modifying ASOs, and other therapeutics will pan out in the future.
Well, unfortunately, we're out of time. Kat, thank you so much for joining us today. It's always great to get you in here, and we know you're busy, so it's nice to have a chat with you. So if we didn't get to your questions, we will be sure to follow up with you. But again, I wanted to remind everyone to join us on October 21st and 28th for our neurobiology scientific sessions to get a more in-depth look at the ALS studies that Kat was mentioning, as well as learn about some of the other great areas that we're working on. I know, Kat, you'll be presenting on Friedreich's ataxia yourself. So, so we've got a lot of great stuff going on. So thank you, everyone, and we will see you at the next JAX Live.
Thanks.